Hello friends and welcome. Today we are going to talk about Chara. The main objectives of today's deliberation are: number 1, to know about the general characteristics of Chara. Second, to know about occurrence of Chara. Third, to gain information about the range of thallus organization in Chara. Fourth, to find out the morphology and cell structure of Chara. Fifth, to give a detailed insight about the reproduction in Chara. And finally, we shall gain knowledge about the life cycle of Chara. To begin with, let me give you a brief introduction. Chlorophycy is characterized by the presence of chlorophyll A and B, starch as reserve food, pyronoids which are surrounded by starch sheath, cellulosic cell wall. The thallus ranges from single unicellular motile form to complex heterotrichous form. Most of the green algae are aquatic, about 90% of which are freshwater. Chlorophycy have been divided into 9 orders: volvocales, chlorococcales, eulotrichales, cladophorales, chetophorales, oidogonales, conjugales, siphonales, and charales. Sexual reproduction occurs by isogamy, anisogamy, and oogamy. The sex organs are usually unicellular. Zygote represents the diploid phase. The life cycle in different genera may be haplontic, for example, Chlamydomonas; diplontic, for example, Codium; isomorphic, for example, Cladophora; or heteromorphic, for example, Urospora. Chara is submerged aquatic alga consisting of an erect branched axis attached to the substratum by multicellular rhizoids. The plant grows to a height of about 5 to 13 cm and resembles in appearance to a miniature horsetail. The plant body is differentiated into nodes and internodes. From each node, a whirl of short laterals may be limited or unlimited. Chara can appear to be a plant that would produce flowers and seeds, however, it is actually a multicellular macroalga. Chara grows attached to the bottoms of ponds, lakes, rivers, and ditches, and can form submerged beds of vegetation. Individual plants can range in size from a few inches in length to several feet in length. Chara has whorls of 6 to 8 branchlets that arise from the nodes along the stem. Monoecious and dioecious species exist, but vegetative plants persist year-round. During times of reproduction, dark, ball-like sporangia appear seed-like along the branchlets. Chara is also known to have strong garlic odor. Once identified by smell, it will be remembered. This macroalga has no true leaves, only branches and branchlets are present. Now coming to the classification of Chara, it belongs to division Chlorophyta, class Chlorophyceae, order Charales, family Characeae, and genus Chara. Now let's find out the general characteristics of the Chara. The plant body is calcified and commonly called as stonewarts. Second, the plant body consists of an erect axis differentiated into nodes and internodes with lateral growth at the nodes. Third, the internodes consist of single undivided elongated cylindrical cell. Fourth, the node is made up of transverse layer of short cells. Fifth, the growth takes place by single dome apical cell. Sixth, it is attached to muddy or sandy bottom by means of rhizoids. Seventh, the reproductive organs are complex and are surrounded by a layer of sterile tissue. Eighth, the motile male gamete antherozoid is an elongated biflagellate spiral structure. Ninth, the female sex organ or the oogonium is a large, oval body covered with multicellular envelope and is known as nucleus. 10th, the zygote germinates into a protonemal stage. Now let's see occurrence of Chara. Chara is represented by about 188 species, out of which 30 species are found in India. Chara plants are often coated with calcium carbonate and hence are commonly called as stonewoods. The plant body of Chara is encrusted with calcium and magnesium carbonate, especially on the plants growing in heavy water. Chara is freshwater green alga found submerged in shallow water ponds, tanks, lakes, and slow-running water. Chara baltica is found growing in brackish water, and Chara fragilis is found in hot springs. Chara is found mostly in hard fresh water, rich in organic matter, calcium, and deficient in oxygen. Chara often emits disagreeable onion-like odor due to presence of sulfur compounds. Chara heti grows trailing on the soil. Chara nuda and Chara grovesi are found on mountains. Chara wallichi and Chara lidropets are found in plains. Now coming to the morphology of Chara. Chara or muskgrass is a submerged aquatic alga consisting of erect branched axes attached to the substratum by an elaborate system of multicellular rhizoids. Plant body is erect, slender, and flexuous. The erect system is differentiated into well-defined nodes and internodes. Nodes consist of several small isodiametric cells in a cluster of 2 central cells. In Chara, single central internodal cell is surrounded by a cortex of smaller elongated cells, half of which are derived from node above and half from the node below. Internodal cell is several times longer than the node. The multicellular rhizoids with oblique septa developed from the lowest node are not differentiated into nodes and internodes. Their functions are in encouragement and absorption. Growth of main axis and its branches is initiated by a single large dome-shaped apical cell, which cuts off segments towards the posterior or bottom face of the cell. Each segment divides transversely into a biconcave upper nodal initial cell, which divides and redivides to give rise to a node, and a biconvex lower internodal initial cell, which remains undivided but elongates considerably to form the internode. Now let us see the range of thallus organization in Chara. The thallus of Chara is branched, multicellular, and macroscopic. The thallus is normally 20 to 30 centimeters in height, but often may be up to 90 centimeters to 1 meter. Some species like Chara heti are small and may be 2 to 3 centimeters long. The plant in appearance resembles equistem. Hence, Chara is commonly called as aquatic horsetail. The thallus is mainly differentiated into rhizoids and main axes. Now first we will discuss rhizoids. The rhizoids are white, thread-like, multicellular, uniseriate and branched structures. The rhizoids arise from rhizoidal plates, which are formed at the base of main axis or from peripheral cells of lower nodes. The rhizoids are characterized by the presence of oblique septa. The tips of rhizoids possess minute solid particles which function as statoliths. The rhizoids show apical growth. Rhizoids help in attachment of plant to substratum, that's mud or sand, in absorption of minerals, and in vegetative multiplication of plants by forming bulbils and secondary protonema. Now, main axis. The main axis is erect, long, branched and differentiated into nodes and internodes. The internode consists of single, much elongated or oblong cell. The internodal cell in some species may be surrounded by one cell thick layer called cortex, and such species are called as corticate species. The species in which cortical layer is absent are called ecorticate species. The node consists of a pair of central small cells surrounded by 6 to 20 peripheral cells. The central cells and the peripheral cells arise from a single nodal initial cell. From the node arise these types of appendages. First, branches of limited growth. These branches arise in whorls of 6 to 16 at the nodes of the main axis, and branches of unlimited growth. They stop their growth after some time. They are also called primary laterals, leaves, or branchlets. Reproductive structures are born on these branches. Number second, branches of unlimited growth. These branches arise from the axils of the branches of limited growth. They also bear nodes and internodes besides a whorl of primary laterals at the node. They grow indefinitely. Third, stipulodes. The basal node of the branches of limited growth develop some unicellular outgrowths called stipulodes. Main axes show apical growth. The plant is attached to the substratum by a well-developed rhizoidal system. Fourth, cortex. Many species of Chara, for example, Chara aspera, Chara infirma, have internodal cells of main axis encapsulated by cortex cells. Such species are called corticated species. The cortex consists of vertically elongated narrow cells. The internode up to half of its length is covered by corticating filaments developed from upper node called descending filaments. The lower half of internode is covered by filaments developed from lower node called ascending filaments. The ascending and descending filaments meet at the middle of internode. The species without cortex, for example Chara corallina, are called ecorticated species. Now coming to the cell structure of Chara. The main axis of Chara consists of mainly two types of cells. Number 1, nodal cells. Number 2, internodal cells. The nodal cells are smaller in size and isodiametric. The cells are dense, cytoplasmic with few small ellipsoidal chloroplasts. Nucleus is single, located towards center of cell, and a number of small discoid chloroplasts without pyrenoids are distributed throughout the cytoplasm. The central vacuole is not developed. Instead, many small vacuoles may be present. The cytoplasm can be differentiated into outer exoplasm and inner endoplasm. In the internodal cell, the central region is occupied by a large central vacuole with the small chloroplasts arranged in well-defined longitudinal series of peripheral cytoplasm. It divides mitotically to form a large number of nuclei in the lining layer. The cytoplasm is also differentiated into outer stationary exoplasm, also known as ectoderm, and inner endoplasm. The endoplasm shows streaming movements. The cell walls between the nodal cell and the internodal cells are porous to help in cytoplasmic continuity between cells. Now coming to the reproduction in Chara. Reproduction in Chara takes place by vegetative and sexual methods. Asexual reproduction is absent. First of all, let us see the vegetative reproduction in Chara. Vegetative reproduction in Chara takes place by different methods. First, bulbils. The bulbils are spherical or oval tube-like structures which develop on rhizoids or on lower nodes of main axes, for example, Chara baltica. The bulbils on detachment from plants germinate into new thallus. Second, amylum stars. In some species of Chara, for example, Chara staligena, on the lower nodes of main axis develop multicellular star-shaped aggregates of cells. These cells are full of amylum starch and hence are called amylum stars. The amylum stars on detachment from plants develop into new Chara thalli. Third, amorphous bulbils. The amorphous bulbils are a group of many cells irregular in shape which develop on lower node of main axis, for example, Chara delicatula, or on rhizoids, for example, Chara frangifera and Chara baltica. The amorphous bulbils are perinating structures. When the main plant dies under unfavorable conditions, these bulbils survive and make Chara plants on return of favorable conditions. Fourth, secondary protonema. These are tubular or filamentous structures which develop from primary protonema or the basal cells of the rhizoids. The secondary protonema, like primary protonema, form Chara plants. Now we'll see sexual reproduction in Chara. The sexual reproduction in Chara is highly advanced oogamous type. The sex organs are macroscopic and complex in organization. The male sex organs are called antheridium or globule, and the female oogonium or nucleus. Most of the Chara species are homothallic. That is, the male and female sex organs are born on the same nodes, for example, Chara zelanica. Some species, for example, Chara wallichi, are heterothallic. That is, male and female sex organs are born on different plants. The sex organs arise on the branches of limited growth or primary laterals, the nucleus above the globule. The development of globule and nucleus takes place simultaneously, but in some species, globule matures before nucleus. Globule. The mature globule is large, spherical, stalked, red or yellow structure. The wall of globule is composed of 8 curved plates, the shield cells, which are joined end to end, giving pseudocellular appearance. Now let's talk about development and structure of the globule. The early development of globule and nucleus is similar. The peripheral cell of the lower node of the primary lateral divides periclinally to form an inner cell. The outer cell functions as antheridial initial and the lower cell again divides by a periclinal division. Out of these 3 cells formed, the lowermost functions as internodal cell, the middle forms basal node, and the uppermost functions as the antheridial initial. The middle basal node cell divides to make 5 peripheral cells. Out of these 5 peripheral cells, the upper one develops into oogonium, 2 lateral ones form unicellular bracteoles, and 2 lower ones, one on either side of oogonium, forms cortex or remains non-functional. The antheridial initial divides by transverse division to make basal pedicel cell and a terminal antheridial mother cell. The pedicel cell does not divide further and forms a pedicel or stalk of mature antheridium. The antheridial mother cell enlarges and divides by 2 successive vertical divisions at right angle to each other to make a quadrate. All these 4 cells divide by a transverse wall to make 8 cells or octet stage. Each cell of the octet divides periclinally and forms 2 layers of 8 cells each. The cells of inner or outer layer divide periclinally to make 3 radial layers of 8 cells each. The outermost 8 cells enlarge laterally to form a curved plate of 8 shield cells. The cells of the middle layer elongate towards center to make 8 rod-shaped manubrial cells. The cells of the inner layer function as 8 primary capitulum cells. Each primary capitulum cell divides to form 6 secondary capitulum cells. Sometimes the secondary capitulum cell divides to make tertiary capitulum cells. Each capitulum cell divides repeatedly to form 2 to 4 long, multicellular branched or unbranched antheridial filaments or spermatogenous filaments. The antheridial filament has up to 250 uninucleate cells. These cells function as sperm mother cell, and each cell gives rise to a single spirally coiled uninucleate biflagellated antherozoid. The mature globule thus is made up of 8 curved sheared cells, 8 elongated manubrial cells, 8 centrally located primary capitulum cells, and 48 secondary capitulum cells. The secondary capitulum cells give rise to many antheridial filaments. Each sperm mother cell forms a single biflagellated antherozoid. Liberation of antherozoids. At maturity, the sheared cells of antheridium separate from each other, exposing antheridial filaments in water. The sperm mother cell gelatinizes to liberate the antherozoids. Now we will talk about the nucleus. The nucleus of Chara is large, green, oval structure with short stalk. It is born at the node of the primary lateral. It lies just above the globule in homothallic species. Now, development and structure of nucleus. The upper peripheral cell of the basal node of the antheridium functions as the oogonial initial. The oogonial initial divides by 2 transverse division to make 3-celled filament. It has lower pedicel cell, middle nodal cell, and the upper oogonial mother cell. The pedicel does not divide further and makes pedicel of the oogonium. The middle nodal cell, under many vertical divisions, makes 5 sheath cells or peripheral cell which surrounds the central cell. The central cell does not divide and functions as the node of the oogonium. The oogonial mother cell elongates vertically and divides by transverse division to make lower small stalk cell and an upper large oogonium. The oogonium contains uninucleate ovum or egg. The peripheral cells or 5 sheath initials elongate and divide by transverse division to make 2 tiers of 5 cells each. The 5 upper tier cells from coronary cells form the corona of the nucleus. The 5 lower tier function as tube cells. The tube cells elongate and get spirally twisted in clockwise direction on oogonium. The nucleus migrates on lower side and receptor spot develops at the tip of oogonium. Large amount of starch and oil get deposited in oogonium. The oogonial cell possesses a single large egg or ovum. Now let's talk about the fertilization. When the oogonium is mature, the 5 tube cells get separated from each other, forming narrow slits between them. Antherozoids are chemotactically attracted towards ovum. The antherozoids enter through these slits and penetrate gelatinized wall of the oogonium. Many antherozoids enter oogonium, but one of those fertilizes the egg to make a diploid zygote. The zygote secretes a thick wall around itself to make oospore. Oospore. The mature oospore is hard, oval, ellipsoid structure, which may be brown, for example in Chara infirma, black, for example in Chara corallina, or golden brown, for example Chara flauda. The oospore inside contains a diploid nucleus and many oil globules in cytoplasm. On maturity of oospore, the inner walls of tube cells get thickened, suberized and silicified. The oogonial as well as oospore walls become thick. The oospore nucleus moves towards the apical region. In advanced stages, the outer walls of the envelope or sheath cells fall off and the inner parts remain attached to the mature oospore in form of ridges. Now let's talk about germination of oospore. The oospore germinates when favorable conditions appear. The diploid nucleus present in the apical colorless region divides by meiosis, forming 4 haploid daughter nuclei. At this stage, a septum divides oospore into 2 unequal cells. The upper smaller apical cell contains a single nucleus and the large basal cell contains 3 nuclei. The 3 nuclei or basal cell degenerate gradually. The oospore apical cell divides by longitudinal division to make a rhizoidal initial and protonemal initial. The rhizoidal initial shows positive geotropism and forms primary rhizoids. The protonemal initial shows negative geotropism and forms primary protonema. The primary protonema differentiates into nodes and internodes. The peripheral cells of the basal node give rise to rhizoids and secondary protonema. The peripheral cells of the upper node give rise to lateral branches. Now finally, we'll talk about the life cycle of Chara. The plant body of Chara is haploid. The vegetative reproduction takes place by the formation of amylum stars, bulbils, and secondary protonema. Asexual reproduction is absent. The sexual reproduction is advanced oogamous type. The male and female sex organs are globule and nucleus respectively. After fertilization, a diploid spore is formed. At the time of germination, diploid oospore nucleus divides to make haploid nuclei and haploid Chara plant. Thus, the life cycle of Chara is predominantly haploid type. With this, we conclude today's lecture. Hope this was helpful. Thank you.